Okay, so good afternoon, members of the SPN community. It's my great pleasure to welcome you all to this seminar. A warm welcome to our two guest speakers who will be sharing their knowledge with us today. So each presentation will be followed by a Q&A session. I encourage you all to actively participate and make the most of this opportunity to engage with Carina and Quendera. And once again, thank you all for being here. And without further ado, Carina, let's get started.
Thank you, Daniela. Hi, everyone. I just wanted to start by praising the Junior Committee of SPN for organizing these events. I wish you the best of luck for the every future events. And thank you for inviting me to give a small presentation of the work that we've been developing here. So today I'll try to show you a little bit on the work that we've been doing on understanding the role of the nucleus accumbens, which is part of the ventral striatum, in encoding associative learning. And I don't know if you are aware, but associative learning is a very basic process, that it's just a type of learning in which one thing will predict another thing. So basically individuals will have to learn to associate something. It can be a stimulus in the environment, it can be a visual, auditory, olfactory cue with a reward, or something that could be punishing for them. And although this is a very simple process, it tends to be a bit complex in terms of neuronal encoding. And this, I'll try to guide you through now how this happens. Because initially, individuals will basically have natural responses to everything that surrounds them. And for example, I'll give you a very simple example in which, for example, a mouse can be in nature or in your house it tends to get there. And if it's hungry, you'll try to forage and look for food. And eventually, if it finds food, it will consume it. And this food will be an unconditioned stimulus to the animal, and it will be pleasurable to the animal. And the animal will tend to learn things about this reward. For example, if it's a cheese, the animal will recognize the smell of the cheese, the taste of the cheese. But on the other hand, imagine that and it will consume it, and this will send a positive signal to the brain. On the other hand, if the same animal is in nature or in your neighborhood, and eventually if it sees a cat, which is a predator, but if it never saw it before, it will understand that the cat will be a threat to its survival. And so what will happen is that the mouse will try to escape as fast as possible. And so this negative signal will be actually sent to the brain, and the animal will probably learn that, for example, the smell of the cat, or if it sees it, a visual cue that may predict the presence of the cat is actually negative to its survival. But on the other hand, as learning progresses and the number of exposures of the animal increases to these stimuli, actually the animal will be able to eventually predict the potential presence of the reward or the aversive signal. Because, for example, in the case of the cheese, if the animal is hungry and it smells something that is similar to cheese, he knows that he'll actually can seek for it, and this will be again a positive signal to the brain, and this will also result in approach behavior. So the animal will actively look for the reward. On the other hand, if the animal, and this will tend to result in approach, as I was saying. On the other hand, if the animal is in its environment, but if it smells the smell of the cat or cat hormones, what will happen is that this will send a negative signal, and the result will be an escape behavior, even if the animal didn't see the cat. But because this is he knows that it's a predator, it may result to a negative event for it. It will try to do everything to avoid it. And so this, from now on, will be a learned response in which everything or anything that reminds the animal of the cheese or the cat will become a conditioning for the animal. And so these cues will turn into conditioned stimuli that predict their unconditioned stimuli. And so this process, although very basic and every animal has it, it requires the engagement of several brain regions that are part of the brain reward circuit. Let me just try to switch here to the pointer. That include both cortical and limbic structures. And these, all of these structures will actually work in a way that in which environmental stimuli that can either predict a reward or an aversive event or a threat will result in actions that the animal will have to perform to either obtain a reward or avoid this threatening behavior. And among this complex circuit lies the nucleus accumbens, which is here. It's a ventral portion of the striatum, as I was saying, and it's basically known to be a limbic motor interface because it integrates cortical signals and also any glutamatergic signal, and it decodes these signals to produce actually motor behaviors. And throughout the years, what is interesting is that dysfunction in the nucleus accumbens activity, and particularly a decrease in its activity, it's actually known to result in decreased motivation to obtain natural rewards, also increased negative states, so lower motivation to obtain these positive, what were before positive rewards, and also low levels of pleasure known as anhedonia. And these are all hallmarks of depression, for example. Thus, it is extremely relevant for us to understand how the nucleus accumbens encodes this signal so that we then can understand better disease. And the nucleus accumbens is mostly composed, which about 95% of its neurons are GABAergic in nature, and they are medium spiny neurons or medium projecting neurons. And they are generally divided into those that express either dopamine receptor D1 or dopamine receptor D2. In addition to this genetic identity, they are also segregated in terms of circuit because D1 MSNs, they send signals to output brain regions, for example, such as, I have your faces in front of me, so I have to switch again this. Sorry, I can't, it's okay. So they send signals to the output brain regions, such as ventral tegmental area, directly from the nucleus accumbens, or they can do this through indirect projections to intermediate brain regions, such as the ventral pallidum. On the other hand, D2 MSNs can only send signals to output brain regions indirectly through sending projections, for example, to the VP. And very classical studies have been focusing, and electrophysiological studies have been focusing on in understanding the role of medium spiny neurons in the nucleus accumbens in response to both appetitive and aversive stimuli. And classical studies have shown in which intraoral administration of either appetitive stimuli, for example, sucrose, or aversive stimuli such as quinine, which is bitter to the animal, so it really doesn't like it, has shown that medium spiny neurons, independently of being, not identified as D1 or D2 positive, actually respond to both appetitive by decreasing activity or aversive stimuli by increasing activity in the nucleus accumbens. Thus indicating that this brain region is somehow relevant for encoding appetitive and aversive events. However, as I said before, medium spiny neurons, they are segregated generally by its dopamine receptor expression into D1 or D2, but these classical studies did not approach these differences. However, more recent studies using optogenetics have actually shown that it seemed like G1 neurons would be more involved in increasing motivation or in appetitive behaviors. And this was a conditional place preference for cocaine, in which activating D1 neurons would increase preference for the cocaine side. And on the other hand, activating D2 neurons would actually result in the opposites. So thus suggesting that D1 neurons would be associated with reward and D2 neurons associated with aversion. But more recently, we've also applied optogenetic tools in our studies, and we actually showed that applying a brief optical activation to both D1 or D2 neurons would result in increased preference for one side of the chamber, so when the animals, in the side where the animals were stimulated. However, when we apply different activity protocols, so with a more prolonged activation of these neurons, we could actually see the opposite, which is kind of an aversive effect. So thus leading us to believe that actually D1 and D2 MSNs could potentially encode both reward and aversion, depending on the stimulation pattern, so meaning maybe depending on the environment. And so here in this study, our main question was to actually try to understand how can actually these D1 and D2 MSNs in the nucleus accumbens encode positive and negative valence stimuli, but also how do they contribute for a cue outcome or for the development of cue outcome associations, meaning associative learning. So to do this, we decided to use a calcium imaging approach. So we took advantage of a genetically encoded calcium indicator that we injected in the nucleus accumbens, and we injected these in both G1 or A2A, meaning D2 mice, and we expressed these under depending on CRE. So we could image in animals both only D1 MSNs and in A2A animals only D2 MSNs. And we can actually follow the activity. So in freely moving mice, we then implant a lens, and we can attach miniaturized microscopes, and so that the animals can actually freely move and perform behavior and receive rewards and aversive stimuli as we wish. And actually what we can do is then to compare the activity of the same neurons in a session, for example, in which they were receiving a sucrose with a sucrose reward, which is highly positive for the animals, and to the activity of neurons of the same neurons in a shock session. And we can actually then overlap and see how the activity of these neurons changes, or if actually it's similar in response to an appetitive or an aversive event. So what was interesting, and I'll show you just both data at the same time, because what was interesting right away to us was that actually the proportion of signals that we had from D1 and D2 MSNs were pretty similar. So what you're seeing here is in red is the proportion of excited neurons to here US1 or unconditional stimulus 1 would be sucrose, and US2 would be food shock. And actually with the proportion of excited, which is in red, or inhibited, which is in blue, neurons, D1 and D2 neurons is very similar in response to both US1 or in response to US2. However, as you can appreciate, in response to a sucrose, most of the neurons in the accumbens respond with inhibitions, while in response to food shock, most of the neurons will respond with excitations. But still, this would just let us know that there are neurons in the accumbens that respond to appetitive and to aversive stimuli. But if we actually wanted to know if the information that these neurons contain, its activity, could actually be used to predict or to really encode appetitive or aversive signals. So what we did to try to answer to this question was to apply a computational model called linear SVM. And so what we do here is that we input the population activity in response to both sucrose and to food shock, and to try to see if actually the model can predict if the activity of X population, of this population, is in response to sucrose, or if the activity is actually in response to food shock. And actually what we saw, and then we can, what we extract out of this is actually the accuracy that these neurons or this population has in being able to actually differentiate between what's a sucrose session and what's a food shock session, for example. And as you can see here on the left for D1 neurons and on the right for D2, we actually see that we have high accuracies of decoding these signals, which means that both D1 and D2 neurons contain sufficient information to actually differentiate between a stimulus that is positive or has positive valence and a stimulus that has negative valence. And so I started by saying also that we are interested in associative learning. So what we did next was actually to expose the animals to an associative learning task, which is pretty similar to what I introduced in the beginning. So basically what we do is that we expose mice through several days to a cue in which, in our case, it's a compound of a tone and the house light being turned on, and 10 seconds or at the 7 seconds after the beginning of the cue, the animals will receive a droplet of sucrose in the box. And we do this while we are recording the activity. So we see here the tone and the house light turning on, and eventually the reward will be there, and the animal will move towards the poke to receive the reward. And again, looking at the proportions of neurons that were activated across learning. So we did 10 days of training, and I only have here the data for day 1, day 5, and day 10. But again, if you look at the proportions of excited in red or inhibited neurons in response both to the cue and to the reward, they are pretty similar between D1 MSNs and D2 MSNs, which shows that basically these neurons seem to have a similar relevance for encoding these associations. Again, so this is what I just said before. Again, however, we wanted to take advantage of calcium imaging. One of the advantages of calcium imaging is that we have a field of view. So we are recording from the same field of view throughout days. And one thing that we can do, which is it can give you a lot of information about how the learning process occurs, is actually to follow single neurons from the beginning towards the end of the task and see if actually the activity of these neurons is constant, or on the other hand, if it could change across time. So we did tracking of the same neurons across the 3 days of learning that I've showed you before, and we analyzed the activity of these tracked neurons in the end. What we did was actually to check at the individual neuron level if a neuron would maintain its activity across time or if it would change. And we did this basically by calculating the accuracy with which a neuron would actually decode the CS and the US, so the cue and the outcome. And so we applied, in this case, we applied the single neuron computational model, because here we wanted actually to follow a neuron from the beginning to the end of learning. And what was interesting, and this is a map of accuracies. This is very similar to what you saw before, but this is just color-coded. So basically what we find, and just in a very simple manner, is that we have neurons here in red or orange that have high accuracy of decoding. This means that these neurons on this day, they can actually distinguish the cue from the reward, right? But this is true for day 1, but we also have a lot of neurons that are actually really bad at distinguishing the cue from the reward. So basically means that these neurons, they do not encode this association or do not seem to encode this association. And then if we follow the accuracy from day 1 to day 5 and to day 10, we actually see that even the neurons that had like a good accuracy in the first session, they lose it, and other neurons begin to be better at distinguishing the cue from the reward. So basically, this seems to show that both of these populations, they evolve their activity or they drift their activity at the single cell level for learning to occur, basically. But we can't forget what I showed before, which is, okay, we have these neurons, they don't seem to be very good individually at identifying the cue and the reward, but we still had like this, right? We had similar proportions of excitations and inhibitions across days. And this is true both for the cue and for the reward. So again, we decided, let's look again at the population. And let's try to see if by inputting the whole population, not each neuron at a time, but inputting the whole population, if these patterns of activity are actually relevant for encoding these cues and these outcomes. And indeed, what we see is that at the population level, there is high accuracy in identifying the cue that predicts this reward, or in this case, this sucrose. So what we show is that actually this pattern of excitation and inhibition within these two populations seem to be necessary to be maintained across days for learning to occur. Having these results in mind, we also wanted to compare these with an aversive stimulus. So because we questioned ourselves, and because literature was saying that activation of D2 neurons is more related with aversive behaviors, we wanted to check if this could be actually the case. So we did an associative learning task, a fear conditioning task, in which the animals would hear the cue, 10 seconds later, they would receive a food shock. And we again did the same analysis. And in terms of population activity, we only have one session. So what we did was that we segregated between initial trials and late trials, which we try to associate with early and late phases of learning. And what we see is actually, again, stable responding across the learning process, and at the population level, as we did for appetitive behavior, we still see high levels of decoding accuracy for both D1 and D2 populations overall. So I hope that until now, what I was able to show you or convince you is that both D1 and D2 populations, they show responses to both appetitive and aversive associative learning, with these populations showing a specific excitatory inhibitory pattern that, although it is maintained across learning, it actually drifts, and other cells will be excited and inhibited, although the total proportions will be maintained for, and this excitatory inhibitory balance will still be necessary for this appropriate acquisition of these CES-US associations. So we asked, and it will take me just one more minute. So from now on, what we were started to think was that, okay, what is the difference between these populations, right? Because these they are not the same neurons. We already know that the ones that are responsive at early phases are not the same that are responsive at late phases. We saw this. And so what could be distinguishing? And because literature has been saying that actually D1 and D2 neurons, they are not so stable, so they might have differences among them that can be genetic. One thing that one approach that we are taking is actually to look at these neurons and try to identify other genetic markers that further distinguish these populations. So we are exposing mice to appetitive and aversive stimuli, and we are processing the nucleus accumbens tissue for single nucleus RNA sequencing. Just to go really fast on this, because I want you to ask me questions. So what we've done so far, and we are just starting on this one, we have been able to identify different cell populations within the nucleus accumbens, which include clusters that are clearly D1 receptor expressing or D2 receptor expressing. In addition, we have looked closer to these neuronal clusters. And as you can appreciate throughout this list, we were able to identify some of them as different clusters being either D1 positive or D2 positive. And so we found 7 clusters that are D1 positive and 5 clusters that are D2 positive. And actually using literature data, we actually were able to find specific transcripts or genes that can actually segregate these subpopulations. This is both true for D1 and for D2. So what we want actually to do now in the future will be to use these distinct genetic identifiers to further target these subclusters or these subpopulations of D1 and D2 MSNs to actually try to understand if there's a differential contribution of these populations to the learning process. We are still at the beginning, we are still exploring this data, but I hope that this will bring a lot in the future. So as conclusions, I'd just like to say that we have been able so far to show that D1 and D2 populations, they equally contribute for a positive, appetitive and aversive learning. And this went a little bit against the classical studies, but it goes towards in line with the data that is now coming out, mainly with calcium imaging analysis. And we also showed, interestingly, that at the single cell level, we see a drift in activity of neurons across learning process. And we also showed that nucleus accumbens contain these distinct neuronal subclusters that may or may not differentially contribute for associative learning. And just to conclude and try to connect this again to the introduction, looking at the future, one of the things that we may be able to take out of this is that if we actually find that there are subpopulations within this brain region that are more relevant for associative learning, these may also be relevant in the context of diseases such as depression, which is the example that I gave in the beginning, because maybe we are taking the wrong approach in treating these disorders and doing like an overall application of SSRIs or dopamine agonists or antagonists, while probably there are subpopulations that are more affected in the disease than others. And the genetic information of these populations could be used for future treatments. So lastly, I'd just like to thank the people from the lab. So this work is being co-led by Ana João Rodrigues. All of the imaging data was collected by Ana Verónica Domingues, who is a very talented PhD student here in the lab. She's about to finish her PhD, actually. All the computational data and data analysis was done by Tauan Carvalho, who is a postdoc here, and all the single nucleus RNA sequencing data or the tissue processing was done by Natasha Gaspar, who is a PhD student, and we are doing the analysis in collaboration with Christian Conrad and Agata Razueska from the Berlin Institute of Health and Charité. And of course, all the internal members are collaborators and also funding agencies for funding our work. Thank you very much.
Well, thank you very much, Carina, for your presentation. So who would like to start with the first question?
I can start if nobody wants to start. Carina, thank you very much. It was really a beautiful presentation and amazing data. I have two questions. One, do you think, so the different populations of cells that are triggered between the two types of learning, could this have to do with the kind of circuitry that it underlies the different responses? So like, for example, with the amygdala for the aversive and for the food, different brain region. I don't know if you thought about this. And the second question, maybe the first one first, and then I can ask.
Okay, so we get this question a lot, and actually we are really interested in doing an approach in which we are targeting specific neurons that receive specific inputs. Amygdala would be our first choice, actually both for aversive and appetitive events, although it's highly associated with aversion and fear behavior. And we, in addition to the inputs that these populations are receiving, we are also interested in the outputs, because one thing that could be happening is that initially the activity of these neurons may be related with the different inputs that they are receiving, which could be from cortical or whatever glutamatergic signals and dopaminergic that they are receiving. But towards the end, maybe what would be actually more relevant is the outputs where these signals are being sent off to. And we are actually interested in looking at the activity of these populations, both at the input and output level. Although this gives a little bit of work, and we are having some technical issues with infecting these cells, but yeah, that's in our to-do list for sure.
Thank you. And the second curiosity is, I don't know if you did it, or do you think that neurons respond differently to different degrees of saliency in the learning?
Yes. So we haven't done this. Well, we actually have done this I showed the first US responses, just exposure to a sucrose session in which the animals were there. They were receiving a sucrose reward every once in a while, like every 30 seconds, but this was uncued. We've done the same, for example, for condensed milk, which is also positive. They really like it, but it's slightly different. The taste is different. And also the percentage of the content of sugar is different from the percentage of sucrose that we are using. And we do see slight differences. But what's interesting, or I think more interesting, is actually that we didn't do this yet, but there's a publication in the nucleus accumbens core. I didn't say, but I'm recording from the medial shell, which is one of the subregions of the accumbens. But there's a study in the nucleus accumbens core that actually compares different shock intensities, food shock intensities, and they do see differences in the magnitude of response of these neurons from a high intensity food shock to a low intensity food shock. So yes, I would say that there will be differences in the coding, most likely either in valence or in the salience of the stimuli itself.
Thank you very much.
Thank you.
So no more questions? Okay, so thank you, Carina. So we'll now move to our next presentation.
Hi, is this visible?
Yes.
My name is Quendera. I'm a PhD student at the Systems Neuroscience Lab and Neuropsychiatry units at the Champalimaud Foundation here in Lisbon. I would first like to thank the invitation for this series and wish the best for the future of the series. My talk today is titled Hexxed Towards the Psychophysics in High Dimensions. This might seem a very esoteric title, and but it has to do with a very simple question, and that question is, how do we learn to do things? So Carina just gave us one very detailed approach of one very specific way in which we can learn. I'm having a more philosophical undertone in this introduction, and I'm going to the early 1860s, where Fechner wrote this book called The Fundamentals of Psychophysics. And the elements of psychophysics, while at the time with a less esoteric cover, had laid the foundation for what today is most of behavioral neuroscience and behavioral psychology. In very simple terms, what was proposed is that variables of the world and their manipulation can be reflected in behavior. And if we are measured to, if you are able to measure behavior, then we are able to have, in some types of tasks, direct correlations between what is happening in the environment and what is happening internally in the subject. So, for example, here in the random moving dots task, subjects are presented with dots moving with a motion coherence that the experimentalist can manipulate. And if we look at the responses of the individuals, whether the dots are moving right or left, we can see that they are directly correlated with the motion coherence. And this is a very simple yet elegant observation that generates the so-called psychometric curve that has been used in thousands of studies across the last hundreds of years. So a bit harder question is, how do we learn to do harder things? So we know that these problems have been studied for a long time, these simpler problems. How about harder things, like, for example, learning to walk or to play tennis? There's a reason why we struggle with studying these problems. That's because of the so-called curse of dimensionality. The curse of dimensionality states that when the dimensionality of a problem increases, the volume of the state space increases so fast that the data becomes sparse. This means that the larger your problem, the more data you need, oftentimes growing exponentially in size. So while this very simple problem requires very simple data, a problem like, for example, learning how to play the game of chess requires extreme amounts of data. In fact, chess is been one of the most studied tasks in human history, and yet we are still not fully, we don't fully understand how to solve every single scenario. One of the approach that scientists took early on was to use so-called decision trees, where you map out every single possible interaction. The problem is that the game of chess has more possibilities than atoms in the known universe, famously. So in order to build, it's impossible to build a decision tree that large. Because of this, these types of problems have more recently been tackled by technology that does not require the full understanding of the problem, but instead can generalize by learning the rules of the problem and trying to estimate what or guess what to do next. I'm talking, of course, about artificial neural networks. Since the dawn of AI, artificial neural networks have shown great promise in solving this type of complex problems. One of them has been highlighted by the team at DeepMind, where they were able to create algorithms that not only solve these very complex games like chess or Go, but they even go as far as defeating the best humans to ever play these games. But they seem to do so not exactly in the same ways that humans do. Lake and colleagues showed, for example, that while the performance of humans and these types of algorithms is equivalent, the time that it takes them to reach the same performance is much larger in these agents. And that can be explained by a multitude of factors, but one of them is that we as humans are constantly exposed to priors. We have a lot of experience with the types of games before we are tested on them, and we also have the ability to generalize across different types of games, which these artificial neural networks struggle a bit with. So if we want to use the tools that we have at our disposal so far to study these problems, we can plot on this axis where we are. So on one end, we have these very simple tasks that we can very elegantly model using the fundamentals of psychophysics. And on the other end of the spectrum, we have these very, very complex tasks like chess and math, learn how to speak French, who seem to be very, very hard to model using these simple techniques. So the question that we pose is there a middle term? Is there something in between that we could, where we could leverage the tools of psychophysics to study it, while at the same time retaining the importance that complex tasks have? So colleagues at the Uejima group have tried to do something like quantifying complexity of tasks. This is very important because when we're talking about complex tasks, we first need to know what complex means. And one example is to look at what I mentioned before, the decision tree, and try to see how deep does that tree expand. That's what they did, and they showed that in most studies that are done to study human cognition have been done with significantly small state spaces, as you can see on the second column. However, most of the AI studies have been done on state spaces that are extremely complex. This makes it so that there's a hole and a big leap between the human cognition literature and the AI literature that we were curious to see if we could explore. Now, is the right task already out there? As good scientists that we are, of course, first we started to look into our own task, but it is important to see if there are tasks out there that already meet the requirements that we need. And one of them is actually the traveling salesman problem, where you are asked to draw a trajectory between known cities. This task is very powerful because it is so-called NP complete, meaning that we can parameterize it and parameterize its complexity by arbitrarily increasing the number of cities to connect. However, when we are going to, if we are going to put this task next to an artificial agent to solve it, there are a few things that we as humans have advantages on. For example, we may be familiar with the geography of the certain map we are given. And also, we are limited, on the other end, by the number of cities that exist. If you want to make it extremely harder, we will start to struggle as humans to try and make these connections. So we opted to design our own task that we called Hexxed. That's the title of the talk. And in this task, which is materialized in the shape of a video game, first subjects are shown this screen that you see right here. So I'm going to ask you as an exercise to just, if you were met with this screen and you didn't know what to do, what would you do? What would be the first thing that you would do to interact with the screen? And because I don't want to make you suffer, I'm going to give you the response of what you should do. In this game, you tap the screen that you see in front of you to increase the value of a target. This target on the top is depicted in orange. So you tap the screen and the target in orange grows in size. Your goal is to collect this target as large as possible. Each discrete action that you make, meaning each touch that you make on the screen, increases the value of the target. However, if you make one action too many before you collect it, the value of the target goes down back to 0. What this means is that what you have to do in this problem is figure out and plan ahead how many actions you want to make before the target becomes too large and you lose it, so that you can capture it at its maximum value. Our problem can be formalized in the shape of an MDP. And here what you see is a visualization of the problem. In color, you have, oh, I apologize, I just realized this is inverted, but in bright color, you have maximum value, and in dark color, you have minimum value. As subjects start to explore the task, what you should do is navigate across this possible state space until you collect a target at maximum value. Importantly, there are several paths that you can take to collect the target at maximum value, and both of them can be equally optimal. At the same time, if you are not accurate in calculating how to collect these targets on time, you will lose them, and your value of your actions will go back to 0. So this is a bit hard to wrap our heads around, but the important piece to understand is that our task is parameterizable and its state space is expansible or compressible. So we can go from a very easy trial where we show you one target or one city to all the way to higher and higher difficulties. And all of these state spaces can be not only parameterized, but they can also be visualized, of course, up to 3 dimensions, and then imagined in further dimensions. But the important bit is that from now on, for the rest of this presentation, we will focus on the easiest example to try and give you an idea of the type of analysis that we are doing in this task. That is an example with a single target. Now, as I said in the beginning, due to the curse of dimensionality, we do run into an issue. If we are trying to study a very complex problem, we need a lot of data. Luckily, we have the tools and we have reached a point in time where we have the tools and the technology to do so. And we did so by deploying this task as a video game. We were able to gather data from 12,000 unique players, adding up to 250,000 plays and over 3 million data points just for level 1 alone. This was done, of course, with the help of the science communication team at University of Berkeley and our collaborators and Champalimaud Foundation. And you are free to download the game at hexxed.io, and I will show you the URL later in the presentation too. Importantly, because we are able to parameterize this game and formalize it in the shape of an MDP, we can also feed it to a robot or an AI in its most simple possible shape, meaning that we can have any type of agent that we so desire solve our game in a very similar way that humans are solving it. And for the rest of the presentation, while we did run this on several types of agents, I will show you data from a single convolutional layer plus 3-layer ReLU agent, which I'm happy to discuss in further detail in the end of the presentation if you so desire. We chose this agent because results, while marginally equal to all the other agents, they were the fastest agents at solving the task. Now, if you go back to the exercise I gave you before, what should you do? If you answered to click this gray bar, then you are statistically aligned with most of our players. What I'm showing you here is a plot of the first ever interaction of our players with our data, with our game, sorry. And what you see is that they mostly clicked there. Some of them opted to click this happy face, very little opted to click the sad face. And this is interesting just to see, because even in just a single action on with this task, we can see that there's a huge homogeneity in the human behavior. When we try to show you now how do these agents, be it human or artificial, navigate this MDP that I showed you before, we are showing you here this graph. On this graph, you have an initial state on the left and a state where you lose on the right, and intermediate states where you get rewards of growing value. And what you can see is that artificial neural networks seem to be exploratory when exploring this space when contrasted with humans. Humans seem, if you just look at it with if you just look at it superficially, overexplore this specific piece of the puzzle, which is not coincidentally the piece of the puzzle that leads to higher rewards. If we look at how the reward space is sampled, meaning how many rewards of which value do our agents collect, you can see that our artificial neural networks seem to explore this space quite broadly, whereas humans seem to be very picky at it. We seem to over-explore rewards of value 1 and rewards of value 6, meaning that the intermediately valued rewards seem to be a bit more irrelevant. Now, when we look at this similar type of approach, but across time, we can see what are the probabilities of getting a reward of value X given the reward that you had and the previous time step. So if I just got a reward of 1, what is the likelihood of me getting a reward of 1 in the next time step, for example? What you can see is that artificial neural networks seem to explore the state space in a more or less random way. There doesn't seem to be a lot of correlation. Whereas if we look at how humans perform these tasks, they seem to be what we are calling sticky, meaning that they seem to stay on the same reward they were in the previous trial. And this can be highlighted. This is highlighted by looking at the diagonal of this plot and how it is explored. Now, if we look at the probability of completing a level, meaning of getting enough rewards to meet a certain threshold of our choice, we see that distributions of when subjects complete the levels are quite different. So for humans, they seem to be quite heavily tailed distribution with a focus in the beginning, meaning we solve the game fast. This is indicative and reminiscent of the work of Lake and colleagues I showed you in the beginning. Whereas artificial neural networks, while they reach similar criteria and they solve the task equally well, take a lot longer to do so. Another feature is that if we look at the raw traces of progression of score over attempts, meaning how much score did you accrue over time until you completed the level, there seems to be a slow gradient ascent of value in these artificial neural networks that go from 0 into meeting our performance threshold here, assigned by this dotted line, over a stable growth over time. Instead, if you look at our human data, you will see that they seem to be what we are calling leapy. The scores usually revolve around 0 up until a point where a single subject figures out how to solve the task and quickly grows its rewards, accumulates its rewards rapidly to reach the performance threshold. So with that said, to summarize, I introduced you today a task that is inspired by psychophysics and that hopefully allows us to manipulate and leverage the tools of psychophysics to study very complex problems. We are not alone, of course. The group of Weijima, for example, is studying the task of Connect 4, the game of Connect 4. And the group of Gershman is using, for example, proficient and experts at solving Rubik's cubes to try and address the problem in a similar way we are. We find that humans are picky, sticky, and leapy, and this hints at some sort of tier-based learning is taking place. This is what I talked about in the beginning. We have a bunch of priors about in the environment and about things like tasks that we solve in the first place or maps of cities, and we seem to be using and leveraging that type of behavior to solve the tasks. Whereas the same perhaps cannot be said about these artificial neural networks. And just on a last note, we are making this data set and environments open for everyone to download and contribute to, hopefully, in the near future. With that said, I would like to thank all of my lab and the funding agencies, of course, and the Champalimaud Foundation for hosting us, and of course the invitation to participate in this series. And yeah, the floor is now yours for questions.
Well, thank you, Quendera. So if we don't mind, I'm going to start with the question. So I think we all heard this quote, which is hard work beats talent. So I was wondering if you are able to distinguish different strategies and if you are able to correlate that with already existing data to see if someone with a certain strategy is more able to perform difficult tasks, such as chess or Go.
Mm-hmm. So one of the things that we are focusing a lot of our work on right now is exactly that, is to try and identify what types of different strategies our players are doing in this game. What we can see so far is that there doesn't seem to be a significant benefit to choosing strategy A over strategy B, but instead that most of our players seem to gravitate towards one of those strategies. In this case, the leapy strategy. That's perhaps not indicative of what is the most optimal, but instead indicative of the way in which humans tend to solve problems. Or we could also say solve our particular, our game in particular, right? I don't know if that answers it, but.
Yes, yes. So any additional questions? Well, in this case, thank you so much, Quendera and Carina, for your presentations. And thank you all for being here. And well, have a great week.
Thank
Goodbye.
you. Bye.